This chapter focuses on cells, and this material that's covered in this chapter is probably some of the most important material you can learn if you're going to be taking microbiology next or anatomy and physiology next, because the basis of those classes is cells. This chapter is going to go over cell structure, what cells are made of, their parts, what those parts do. And it's also going to go over very specifically what the plasma membrane is and how the plasma membrane works. That also, all of this is extremely important for those future biology classes and anything that you might learn related to biology. So this introductory slide is showing you a few different kinds of cells. There are so many different shapes and sizes and appearances of cells on Earth that it's unbelievable. So this is just a tiny little taste. But the two upper cells, as I labeled there, are both single-celled eukaryotic organisms that live in water. Those are commonly called protists. And in just one drop of water, there could be hundreds of them in there. The bottom two images are cells from our own human body. So nerve cells that are long and stretched out, used for communication, and muscle cells that are also long, and they contract and elongate and help with movement. So maybe you can sort of sense one of those themes that was in chapter 1 emerging here again. Structure determines function. Every single one of the types of cells that I'm showing you on this slide, just by understanding something about its shape and form and structure, that tells you, informs you how they might work, how they might function. So there's a lot of themes that are coming together here. And there's a lot of new information that's going to be very important as we move forward. You know, most people have heard about cells. In fact, you may have first learned about them a little bit in maybe middle school or maybe high school biology. You learned something about cells. Maybe much of what we cover in this class, or in this particular chapter, I should say, you've heard before. So the idea or the concept of a cell is not anything extraordinary today in the 21st century. But a few hundred years ago, we had no idea what cells were. We had no idea they existed. So like with a lot of things, we've come a long way, and we've developed a lot of new technologies based on our increased knowledge. Just look at the vaccines we're using today to help prevent serious illness and death. So cells were first discovered a couple hundred years ago, way back in 1665. Robert Hooke, who was an engineer in England, had an interest in science and biology, and microscopes had recently been invented earlier in the century. And he prepared a piece of cork that comes from the bark of a tree, and looked at it through a microscope, maybe much like the one that you see in the bottom right-hand corner of the slide, using candlelight. Think about that. No electricity yet. And what he saw, of course, and what he reported scientifically was a revolution. That in that piece of cork that many people are familiar with, that was part of the bark of a tree, there were these microscopic little chambers, microscopic little rooms, and he called them cells, which from Latin, celluli, means rooms. Now, a few years later, 1674, another scientist, Antony van Leeuwenhoek, reported his observations of living cells. He looked at lots of organisms from pond water and elsewhere, did intricate drawings of them, and shared them with the scientific community. So these early scientists, or these scientists anyway, are given credit for the discovery of cells and for showing us and telling us about living cells. Now, a couple hundred years later, or almost, in 1839, what's called cell theory came together. Biologists had been studying or looking at cells for a long, long time, not really appreciating how important they were. And a couple biologists wrote some books, published some papers, and what they wrote about is called cell theory. Cell theory is a list of general principles that describe something important about life. And those general principles include, number one, that cells are the basic units of life. They are where the properties or the chemistry of life takes place. Another way to put it is cells are the simplest collection of matter that can still be alive. Kind of like how an atom is the smallest, simplest particle that shows the properties of an element. Another principle of cell theory is that every single organism, every single living thing on Earth is made of at least one cell. So there are many, many single-celled organisms like bacteria or those protists I showed you on the introductory slide. And then there are many multicellular organisms like us, animals, plants, fungi. But no matter what, every living thing is made of cells. And then finally, the third principle of cell theory is that all cells come from pre-existing cells. They don't arise spontaneously out of dust or nothing. That's what scientists used to think, was that life could just come together spontaneously from kind of random matter in the environment anytime. But after careful observations, scientific reports, nobody had ever been able to report such a thing happening. And at this point, biologists were looking at cells dividing under a microscope and making new cells. So it stood to reason that all cells come from cells that already exist. So these three principles make up what we call cell theory, and they are explaining a lot. And even though they're old principles, 150 plus years old, they still form an important basis for biology today. Indeed, some of the most important themes and concepts you learn in this class are basically rooted in these principles. Now, back to multicellular organisms, we are essentially cooperatives of cells. We are giant colonies of cells that are all cooperating with one another. We're made up of dozens and dozens of different kinds of cells, muscle and nerve and epithelial cells and so on. And each of these different kinds of cells has their own job. This is a relatively new evolutionary event. For example, life started 3.5 billion years ago. We have strong evidence for that. But animals didn't come about until about 600 million years ago. So think about that. For almost 3 billion years on Earth, life existed, and there were no animals. And then when there were animals, they were made up of many different kinds of cells. So anyway, it's a relatively new event. And we have plants as well that are multicellular and made of different cells. So getting at the bottom line of this whole slide, cells are the basis of life. Now, of course, all cells are small, super tiny. I'm willing to bet that you have never seen a cell in your entire life, unless you were looking through a microscope. But that's the point. They're small, they're tiny. There's a good reason for that we're going to get to very shortly. But this figure that I have on the slide, taken from a source outside your book, kind of gives you a sense of the size ranges of cells. Now these metric units, we need to go over what these are and how they relate to each other. I think you may have some experience with metric measurement, but on the very top of the figure on the left, units of length are measured in meters, m for meter. cm is centimeter, mm is millimeter. The little weird U-looking thing, which is actually called a mu, a Greek letter for U, is a micrometer. And then the nm is a nanometer. So that's going from the biggest units down to the smallest units. Now this is a logarithmic scale, so that enables us to show a huge range of sizes in one table or graph. So there's a whole bunch of different things that are shown here to give you a sense of size, with the cells being right there in the middle of the figure. But for example, humans, we're about 1.5 meters, 2 meters tall. The longest cells are almost 1 meter long. Those would be muscle cells. Eggs are pretty big. An egg is a cell, so a bird egg, like a chicken egg, is pretty big, but there's a lot to that cell. Now, the reason why some of these cells, like an egg, can be so big is because they're not doing anything. There's not much metabolism there. So they're not very active. They don't require a lot of exchange of materials with their environment, a lot of energy, so they can be big. But most cells are very active metabolically. Little single-celled creatures living in the water, swimming around. They need a lot of energy. They use it up quickly. They need to move things in and out of their little cells really rapidly. So that forces them to be small. But if you keep looking at that scale, we get down to a human egg that's a little bigger than 100 micrometers. You could just barely see that with your eyes. Most plant and animal cells are between about 10 and 100 micrometers, which means you really couldn't see them with your eyes. You would need a microscope. Some are smaller than that. Most bacteria are between about 1 micrometer and 10 micrometers. And you need a fairly powerful light microscope to see them. And then if we go smaller than that, we can see the smallest bacteria, and then we get into viruses and, ribosomes and molecules and so on. So the overall point is that if you look at this carefully and think about it, you can get a sense of how big cells are. Now, in order to see these cells, of course, we need to use a microscope. And light microscopes magnify things, make them look bigger. When we magnify things, we're not actually making them bigger, we make them look bigger. So we're increasing their apparent size. Now, light microscopes can magnify things up to about 1,000 times before resolution begins to be lost or diminished. Now, resolution is our ability to distinguish two objects that are right next to each other. That's resolution. So our human eye is able to resolve things down to, I think, about 1/10 of a millimeter, about 100 micrometers. It's pretty tiny. And anything that's smaller than that, we cannot resolve. We cannot tell them apart, really see them. So microscopes, light microscopes, by magnifying things, we are able to resolve things more easily and tell them apart. So there are also some new technologies with more advanced microscopes that allow us to actually see things even more clearly. They're getting, pretty fancy. But there's a variety of ways that we can increase contrast when we're looking at specimens in a microscope. Contrast is our ability to accentuate differences, to tell things apart. You might be familiar with contrast if you've ever fiddled around with photos on your smartphone or your computer and use maybe a scale for adjusting contrast. And what contrast does is it makes things kind of stand apart more. We can visually tell them apart more. So I'm going to mention that again on the next slide. Let's keep going. So there's a whole bunch of different kinds of light microscopes. If we were able to meet in lab at GCC, we would be pulling out light microscopes from our cabinets called bright field microscopes. And that's a standard kind of microscope that most labs use, students everywhere use. They can be very fancy and very powerful, but they're pretty basic kinds of microscopes. And basically, we're just passing light through a specimen, a very thin specimen, because light has to go through it. And by doing that, then the light passes through some lenses to help magnify, and then we can see it. But just doing that and nothing else, there's very little contrast. We don't really have the ability to tell things apart very easily. Many cells, for example, don't have colors to them. They're basically almost transparent. So we really can't see many details about them because there's so little contrast. However, part B on here, we can add dyes or stains to our microscope specimens, chemicals that make things become darker or colorized. And we can look at them that way through a microscope, and that helps to increase or enhance contrast. So if you look at the figure there, bright field microscope, stained specimen. So a simple little colored stain was added to the very same cells that you see on the top of the figure. But now you can see like a nucleus pop out in the middle of the cells, that little dark circle. And you can see the edges of the cells and the inside where the cytoplasm would be. So now we have contrast, and now we can see detail. So I'd like you to know about the bright field microscope. I'd like you to know that name. I'd like you to know that with just using light, you don't see much contrast, but by adding dyes or stains, we can see more detail. And that by adding dyes and stains, that usually kills the cells. So there's a trade-off there. If we don't use dyes or stains, we might be able to see living cells and watch them under a microscope. But if we do use dyes or stains, we might kill them. There's other kinds of microscopes that are shown in the figure and that I'm briefly describing here on their slide. And I'm not going to ask you about these other ones. I just ask that you kind of check them out, look at the images and the captions here, and just kind of get an appreciation that there's some fairly advanced technologies to help us see details of cells in different ways. And some of the images that you're going to see in this textbook are taken with these different kinds of microscopes. Now, on the last slide, I was telling you about light microscopes. Here, you're learning about electron microscopes. These are much more advanced technologically. While light microscopes were invented, as you've learned, in the 1600s, electron microscopes weren't really invented until the early 1950s. And whereas we use light to help us see things with a typical microscope, we literally use electrons to see things with electron microscopes. We don't look through them like we do light microscopes, but there's like cameras and computer boards and so on that are picking up images, and we see these images on like computer screens or monitors. So it's a very different kind of microscope operating in a very different way. But fundamentally, what's happening is a beam of electrons is sort of being fired at a specimen. And based on the way those electrons are like bouncing off or passing through your specimen, same thing with light. Light bounces off things or passes through, and that's how we see stuff. So these electrons are bouncing off or passing through, and then there's sensors that pick up the electrons, and then those sensors basically transform that into an image for us. And that's what we see. Now, there's two kinds of electron microscopes. One is called a scanning electron microscope, and it looks at surfaces, and it helps us to get an idea of the three-dimensional shapes of things. The transmission electron microscope, abbreviated TEM, looks at thin slices through some kind of specimen, like a cell. And based on density differences of that specimen, the electrons bounce off in different ways and we see an image. So between these two kinds of microscopes, electron microscopes, we can really get a good sense of what things look like down to a really tiny level. So whereas light microscopes can magnify 1,000 times and have very good resolution, an electron microscope can magnify to almost 1,000,000 times with good resolution. Wow, that's amazing. So look at the figure I'm sharing with you on the slide. The top figure is showing you these little hair-like things that stick out of cells in our body. They're called cilia, and they wave back and forth. You'll learn about them in this chapter. So the scanning electron microscope helps us to see, like we're standing up above them, the three-dimensional shapes of these things. The bottom figure is showing you a thin slice through the edge of a cell, the same kind of cell that has these cilia. And that thin slice is slicing through some of the cilia. So you see these little circles. That's like slicing through noodles. And all those cilia are attached to the cell on the bottom of the figure. You see like the outside edge of the cell. But it's all a thin slice, and by looking at lots of thin slices, we can get a really good sense of how cells are put together and what they're made of. So two kinds of electron microscopes, scanning and transmission. Know these names, these terms, and know what they're used for. So this is kind of fun to show you. This is the same organism. It's called a paramecium. If we were able to meet in lab at GCC and use microscopes, I would show you some live parameciums under the microscope, and you could see them swimming around. Maybe you have looked at them in some other biology class. I hope so. But the point here of the slide is to show you that in the upper left, there is an image of a paramecium taken with an SEM, scanning electron microscope. And man, all that detail just pops out. Those little tiny hairs, those little cilia that are sticking off of it. It's amazing detail. Now the bottom image is taken with one of the types of light microscopes called the differential phase contrast light microscope. And it really makes details pop out inside the cell. We can see the internal structure really well. So again, the only point of this slide is to show you that with different kinds of microscopes, we can learn different things about the same kind of cell. It's pretty powerful. I put a video on here, a link. Watch the video of Paramecium to see them alive. So you can see what these guys actually look like if you were looking through a microscope and could see them swimming around. They're totally harmless. They live in ponds and lakes and things like that. You could swallow them. They wouldn't hurt you. There's all kinds of little microorganisms that live in water like this that are touching you and swimming around you when you're in a lake or a river, and they're completely harmless. They wouldn't do any kind of harm to you. Well, I've tried to send the message to you that cells are really tiny, and we got to use microscopes to see them. But why is it that cells are so small? Is there a reason? Yeah, there is. There's a physical reason, a reason based on physics that has to do why cells are so small. So I'm going to attempt to explain it to you here. So any object, any three-dimensional object has space inside of it, okay? And the space that's inside of an object is referred to as its volume. Volume is the amount of space inside. So if you're purchasing milk in a supermarket and you buy a pint or a quart or a gallon, those are different units of volume. You probably know a quart is more volume than a pint, and a gallon has more volume than a quart. So volume, the amount of space inside. Now, the amount of covering on the outside of an object is called the surface area. Surface area. So with those two things in mind, the space inside is the volume, and the covering on the outside makes up the surface area. Let's now give a reason why cells are so small. As any object of a particular dimension, meaning like a sphere, all spheres have the same dimensions, okay? Whether you're a marble or a golf ball or a tennis ball or a basketball or whatever, or a cube. You could have a whole bunch of boxes nested inside of each other. They're all cubes of different sizes. As any object of a particular dimension gets bigger, this is what happens. The volume, the amount of space inside, increases at a faster rate than the amount of surface area on the outside. So the volume is outpacing the surface area. This is a physical fact, and it can be shown mathematically, like this figure that I'm put on the bottom of the slide is showing you this. So let's take a look at the figure. We've got a little cube, and we've got a big cube, and then we've got a cube that is the same overall size as the big one, but it's made up of a whole bunch of little ones. Okay, so we've got three comparisons here. So we can calculate the surface area of the little cube. We can do the same thing for the big cube. And for all the little cubes put together, we can also calculate the volume of the little one and the big one and all the little ones put together. And after we do that, after we calculate the surface area and the volume, we can compare the amount of surface area to the volume and come up with a ratio, which is on the bottom. So, for example, the little cube, the sides each have a dimension of 1. And here's the formula for surface area. So if you plug the number 1 into that formula, then you get the surface area is 6. Okay, you could do it, or just take my word for it. If you plug that number 1 into the volume equation, then the volume of that little cube is 1. So the amount of surface area compared to the volume is 6 to 1. That's where the number 6 is down here. Now let's do the same thing for the big cube. The big cube has dimensions of 5. So if you plug that number 5 into the formula for surface area, the big cube has a surface area of 150. And you plug that number 5 into the volume equation, the volume is 125. If you compare the amount of surface area to the volume, there's 1.2 times the surface area as the volume. Check it out. As this object, this cube, got bigger, the volume is increasing at a faster rate than the surface area is. And that's why the ratio is decreasing. Now, what's shown over on the far right is you could take an object that's the same size as the big cube, but divide it up into a whole bunch of little cubes, and look what happens. You can maintain that surface area to volume ratio because they're all little tiny cubes. Okay, here's the point related to cells. Because this happens to every kind of object, including cells, as cells get bigger, the amount of volume on the inside increases at a faster rate than the surface area. So at some point, the cells just can't move materials in and out of the surface fast enough to take care of the volume inside that's using all those materials for metabolism. There's a limit. Just can't move stuff in and out fast enough for the cell to still maintain itself as a living thing. So it has to be forced to be small to maintain that surface area to volume ratio. That's why cells are small. Now for us, we're made up of literally trillions of cells, literally trillions of little tiny microscopic cells. So we're basically the equivalent of this cube that's made up of little tiny cubes. We're made of all these little tiny cells that have lots of surface area compared to their volume, and therefore we can be alive. We can move stuff in and out of our cells quickly enough that they can all be alive. That is why cells have a size limit. A big part of it has to do with the surface area to volume ratio. So now let's start learning about the characteristics of cells, the parts of cells. So we'll begin by just outlining what characteristics all cells have, every cell on the entire planet. Whether you're a bacterial cell, a mushroom cell, a whale cell, it doesn't matter. All cells on the whole planet have these 4 characteristics. They have a plasma membrane, they have cytoplasm, they have DNA, and they have ribosomes. So you should know these, and you should know what each of these is all about. So hence I've got descriptions. The plasma membrane. We're going to go over very soon here what the plasma membrane is made of. But for now, what does it do? It's the outer boundary of cells. If you look at the little drawing that I sketched off to the side, the plasma membrane is literally the outer covering or boundary of the cell. And its job is to hold in the cell's contents, like a balloon is holding in air or water. It's the same idea. And that plasma membrane is regulating or controlling what goes in and what goes out. Kind of like a bouncer at a nightclub controls who goes in and who goes out. That's like what a plasma membrane does. Now, the cytoplasm is the interior region of the cell, and it's kind of semi-fluid. It's kind of jelly-like, and the semi-fluid part is called the cytosol. It's made of water, which is the solvent. Remember that term from chapter 2. And organic monomers and ions. Okay, so chapter 3, organic monomers, sugars, amino acids, nucleotides, parts of lipids, glycerols, fatty acids. Okay, so those are all in there on the cytosol. And also ions like sodium, potassium, magnesium, iron, calcium. They're all in there. They're all part of that cytosol in the cytoplasm, the liquidy part. Then there's DNA. And as you've already learned, as you already know, DNA is where a cell keeps its genetic instructions for making proteins and other molecules. And the DNA is found in structures called chromosomes. And we'll come back to that in a later chapter. Prokaryotes and eukaryotes have different shapes of chromosomes. And I'll mention that coming up again. Finally, ribosomes. You've heard of ribosomes. Ribosomes are mostly made of RNA and some proteins. And they're the little factories. Think of them as the protein factories, where the genetic instructions are used to link together amino acids and make polypeptides. And there's some differences between prokaryotes and eukaryotes in terms of the kinds of ribosomes, but that won't be important for us. So you should know this list. You should be able to describe and explain what these parts are. And you should be able to draw a picture, kind of like the one I drew on the slide that labels these parts. If you can do all that, then good. You know the 4 basic characteristics of cells. Now, one of the parts of cells that I mentioned on the last slide is the plasma membrane. And it is so important. Near the end of this chapter, there's more sections about the plasma membrane and how membranes work. I told you at the introduction of this chapter that understanding the material, the concepts, the terms in this chapter is super important as you move on to maybe take microbiology or anatomy and physiology. So you should really get to know the plasma membrane. You should really try to understand it. The plasma membrane is one of many kinds of membranes in eukaryotic cells. And you'll see what I'm talking about in the next section. It's the only kind of membrane that prokaryotic cells have. But as you've already heard, the plasma membrane forms the outer boundary of all cells. It holds in the contents of the cell. And it serves as a selective barrier, like I said, kind of like a bouncer at a nightclub. Hey, you can come in. You're a molecule that can pass through, but you can't come in. Not going to allow you in. You're a kind of molecule that's not allowed. It's that kind of thing. And this shows the property, like in chapter 1, of internal regulation. Remember, that's one of the properties of life, is that living things regulate what's going on inside of them for homeostasis. So the plasma membrane, that bouncer on the outside of cells, has that job of regulating and controlling what goes in and what goes out. So important that you know that. If you look at the figure that I have on the left, in the upper left-hand corner, look, it says TEM of a plasma membrane. What is TEM? Transmission electron microscope. So we're looking at a thin slice on the very edge of a cell. And you can see how tiny it is because there's a little size scale that says 0.1 micrometers. That is insanely small. So you can see there's a little box around that plasma membrane. Okay, and that's what we're learning about. That's what you really need to understand, is what's going on at that outside boundary. Then there's a cartoony kind of figure that's showing you what the plasma membrane is made up of. And you need to really know this. The plasma membrane is made of a phospholipid bilayer, cholesterol, and different kinds of proteins that have different jobs or functions. So we'll come back to that a little bit later in the chapter. But at this point, you should know these 3 molecular components of the plasma membrane. And you can see them all labeled in the figure. Probably be a good idea for you to be able to draw something like that and label it. Now, the phospholipids spontaneously organize themselves. Nothing needs to direct them. Nothing needs to make them do this. They just happens all by themselves. They spontaneously organize into a bilayer, which means 2 layers. So there's 1 layer of phospholipids on one side of the membrane, and then there's another layer of phospholipids that makes up the bottom part of the membrane. So there's 2 layers of phospholipids. And what you can see when you look at the picture, and if you really think about this, why are the phospholipids pointing in the directions that they are? Remember from chapter 3, phospholipids have a hydrophilic head, and they have hydrophobic tails. So what's happening is the hydrophilic heads are pointing towards water on one side of the cell, and hydrophilic heads in the other layer are pointing towards water on the other side of the membrane, like inside the cell. And the fatty acids, the hydrophobic tails, are all hiding in the middle. So this just happens spontaneously. It happens all by itself. The phospholipids naturally organize themselves into those 2 layers because of their physical properties. Now there's a couple proteins that are drawn there in the figure. Make note of the one on the left that has a channel through it. A really important type of protein in our membranes is called a transport protein. And their job is to transport or move molecules or ions in and out of a cell. So many transport proteins literally have a tunnel or a channel going through them for very specific kinds of substances to move through. For example, your textbook talks about aquaporins. Aquaporins, you should know about these. Aquaporins are a specific type of transport protein that only allows water molecules to move in and out of a cell. So a cell can control how much water is moving in or out of it by putting aquaporins in the membrane. If it doesn't have many aquaporins, water is not going to be able to go in and out very much. If a cell has a lot of aquaporins, water will go back and forth a lot. So different kinds of cells in your body have different amounts of aquaporins. That helps to control the flow of water in and out of the cells. So that's about it for now for plasma membranes. You should know what they do, like I tell you up on the top. Know what part of the cell you'd find it in, outside boundary. Know what the membrane is made up of, phospholipids, cholesterol, proteins, and understand why the phospholipids organize themselves in a bilayer. Very important you know that. And know what aquaporins are. So we're going to end part 1 of chapter 4 with a brief overview of prokaryotic cells. If you take microbiology, you'll learn so much more about prokaryotic cells. But this is an introductory class. It's covering lots of material. That's the foundation for what you might study later. So prokaryotic cells include bacteria and archaea. Karyotic means nucleus, and pro means before. So to describe a cell as prokaryotic is to say it doesn't have a nucleus, no nucleus. There's DNA in there, but the DNA is not enclosed inside of a membrane. That makes a nucleus. Only eukaryotic cells have a nucleus. So that's what prokaryotic refers to. Prokaryotic cells are much smaller, typically, than eukaryotic cells. You were told that a few slides back when we discussed the size ranges of cells. So they're very tiny. You need a powerful light microscope to see them. And they're much less complex than eukaryotic cells. They don't have a nucleus, and they don't have many of the separate compartments called membrane-bound organelles inside of them. But their internal chemistry is very complex. They are living things, and the way they live is very similar to the way our cells live. And there's a lot going on in there. So while their structure is relatively simple, their chemistry is pretty complex. Now there's 7 parts to prokaryotic cells that you need to know about. And I've listed them here and briefly say what they are or what they do. So you should know this. If we kind of work from the inside out of a prokaryotic cell, you might say, okay, the very inside has DNA. And the DNA is located inside structures called chromosomes. And this DNA is found in a region of the cell that's called the nucleoid region. That means nucleus-like, kind of fake nucleus, kind of like a humanoid is a fake human. So the DNA is found in chromosomes, and that chromosome is making up what's called the nucleoid region, just the part of the cell where it's located, that's all. Then number 2, that DNA, that chromosome is surrounded by cytoplasm, and in that cytoplasm are ribosomes. Remember, the ribosomes are little protein factories. That's where the genetic code is used to link amino acids together and make proteins. Then when we get to that boundary of the cell that's holding all this stuff in and controlling what goes in and out, that's the plasma membrane. Sort of regulates the traffic. Then if we go just outside the plasma membrane, there's like a shell that surrounds the plasma membrane, and it's called a cell wall. And it's pretty rigid, pretty hard, and that provides shape and structure to a prokaryotic cell. So it's not a flimsy bag of cytoplasm, but it's like, you know, in a box. The cell wall is like a box. whereas the plasma membrane just inside of the box is like sort of fragile plastic wrap, could tear easily. Just outside the cell wall, many bacteria, but not all of them, have what's called a capsule. It's a layer of sticky proteins and sugars that helps the bacteria, the prokaryotes, adhere or stick to surfaces. And it also protects them from dangerous chemicals in the environment that might harm the bacteria. Then you can see little hair-like things sticking out. Those are called fimbriae. In your book, they're called pili or pili. And they are used for attachment, for sticking to things. And then finally, the last thing listed here, flagella. Flagella are very long, hair-like things, and they spin around. They're like propellers, and they help bacteria swim through the water. So those are flagella. Not all bacteria have flagella, but those that do are able to swim and move. So be familiar with these characteristics. Very important. We'll build on them later. And take a note of the figure I have here. Not only am I showing you these parts, but look, there's a cartoony picture, and then there's a TEM, transmission electron microscope. So it's a very thin slice through a certain kind of bacteria, and we can see in there. I put a link to a video to watch about bacteria, so why don't you check that out and kind of get a different perspective than what I'm giving you here on this PowerPoint slide.